Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really fascinating guest involving science, technology, medicine, creating a better tomorrow for many people out there. Today on the show, we have the honor of being joined by Dr. Aliza Apple, who is both Vice President of Catalyze360 AI. This is a really fascinating early stage biotech centric ecosystem at Eli Lilly that's focused on enabling early stage discovery, agnostic of therapeutic area, and as they say, removing a lot of the barriers to promising and emerging science. She also serves as global head of Lilly TuneLab, which there she leads strategy, the build, and the launch of this really interesting external facing artificial intelligence machine learning effort for drug discovery. In previous roles at Lilly, Dr. Apple served as the chief operating officer and head of Lilly Gateway Labs on the West Coast. There she supported the needs of various local biotech companies, the local biotech ecosystem through early engagement and providing tailored offerings to meet their needs. And then prior to Lilly, Dr. Apple served as the co-founder of a really interesting biotech company called Santa Bio. It was a venture-backed precision biologics company focused on autoimmune diseases, as well as an advisor to the founders of Firefly Biologics. And then prior to the biotech world, Dr. Apple was a partner at McKinsey in their life sciences practice. She spent 9 years there, both in New York and San Francisco, collaborating with leaders from all over the place in terms of biopharma strategy, developing corporate and portfolio strategies, enhancing business development. Dr. Apple earned her Ph.D. in bioengineering at UC Berkeley and UCSF. She also holds a Bachelor of Science in Physics from Georgetown University, and she did postdoc work at Cornell Medical College, supported by NIH, courtesy National Research Service Award. A lot of really cool topics to get into today. I'm really excited about this episode. Dr. Aliza Apple, thank you so much for taking time to come on the show.
Thank you. Thanks so much for having me. Really appreciate it.
Yeah, I'm interested in in going into all these topics, especially a lot of what you were doing sort of in the early days, sort of pre-biotech, pre-Lilly. But as I typically do, I love looking at our guests' research history, and in your case, a really an area very fascinating, interesting personally to me, that had to do with not just 3D sort of bioengineering of tissues, but this really intersects, I guess this intersects sort of with your degree in physics, thinking about not just the biology of how stem cells work and ultimately form tissues, but sort of all the interesting external stimuli and signaling that happens between cells that normally only find the embryonic environment. I would love for you just to talk a little bit about your background and a little bit of how this specific dissertation topic fell into your lap, because it's awesome. I love those teams.
Yeah, no, thank you. I appreciate the enthusiasm. I think I probably have a pretty interesting zigzagging path. I choose to see that in a humorous way. But I think for me, I personally struggled considerably with the decision of should I go do a PhD or should I get an MD? And I think that's probably one of my sort of earlier, sources of the zigzag, so to speak. So we had really close family friends that had done quite a bit of regular medical missions with the Helen Keller Foundation doing eye surgery across the world. And they would come home with pictures and stories. And I felt so incredibly inspired by the dream of, frankly, pursuing a career that would have an impact on people's health. And so, in my earliest incarnation, I dreamed of, becoming a doctor, and Doctors Without Borders would be, the sort of epitome of success in terms of how you could really contribute. And as I dug into my undergrad studies, I found myself really drawn to physics and math in particular, and I loved the ability and the skills that it was teaching me from a true reasoning perspective, and really thinking about applied concepts, as opposed to, unfortunately, at the time, a lot of my pre-med classes were just taught with rote memorization, which was a lot less inspiring. So it did push me into this realm of, okay, what can I do that will be interesting, creative, exciting, and have this patient impact that I was really drawn to from the start. As I was getting underway with that question, I actually spent a year as a research associate in Sydney, because of course I had to make it fun. So I moved to Sydney and worked in a microbiology lab where I focused on disrupting bacterial biofilms like cholera and E. coli in particular in drinking water tanks. And so I found that, wow, there is actually really a concrete opportunity to do research that can have an impact that could maybe even have a global health impact. And so it really set me on the course of, okay, research is going to be the right choice for me. And as it came time to really think about applying to grad school, this was when there was so much interest in embryonic stem cells, to your point, but frankly, also a lot of controversy. A lot of controversy around, of course, the sourcing of the cells, but also the funding, and what truly would ultimately be the likelihood of actually taking something all the way to a patient, and realizing the impact with all of that controversy was something really deeply on my mind. I have a very pragmatic, perhaps to a fault, perspective on things. And so with that in mind, I started gravitating much more towards mesenchymal stem cells and regenerative medicine, and began really just reading pretty obsessively about where the field was, what might be exciting for me to dive into in grad school. And in this bioengineering program at UCSF, UC Berkeley, there were a number of labs, particularly on the UCSF side, that had really deep integrations with some of the medical departments. And in particular, I ended up being able to select a lab, Dr. Jeff Loatz's lab, that was actually really closely tied with the orthopedic surgery department. And that is really where a lot of my interests and ideas came from, discussing with surgeons what they were seeing, because we actually had a lot of residents and surgical fellows rotate through our lab, better understanding what the surgical approaches look like today, or at the time. This was already a long time ago. And really better understanding how we could come at it from a tissue regeneration, essentially the early days of thinking about cell-based therapies, of course, very different from how you think about CAR T today, but what could be done from a cell engineering perspective, and frankly, what, again, back to the kind of pragmatic view, what would be scalable, what would be cost effective, how could we actually think about this we weren't using these words at the time, but what would be an autologous cell based approach? And so that's a big part of what I dug into in my thesis work.
Yeah, and it's and again, you continue sort of doing some of this work in your postdoc, looking at really cool stuff on collagen and adhesion proteins and all that. And then, as you were saying, but a zag comes here, because you don't head off and start a tissue engineering company. You actually head further into the world of global public health. And I neglected to mention your time in Sydney and working on cholera biofilms, but next, you head off to Ethiopia and Malawi and Mozambique and a lot of other ports of call, working on vaccines. And again, and then you get to McKinsey, and you're also writing reports on sort of global health at the same time. But Talk about this as well, because obviously it's 2025 right now, and we got all
Yeah.
sorts of vaccine issues, let's just say, going on. But
Yes.
very important you were out there in many strengthening resilience, strengthening these different bio health systems in countries that needed this type of help. So talk about your global public health journey as well.
Sure, yeah. So coming out of my postdoc, I was I was somewhat surprised as a wonderful, naive, freshly minted postdoc to realize that there were very few industry jobs that actually needed any of the skill sets I developed in the lab. So cell therapy, cell engineering was not really a thing yet. Certainly not if you were really looking to go get a job. And I had essentially already decided that I wanted to move away from academia, which I was
Mhm.
deeply grateful to from a training perspective, but that, I wanted to push my career into the private sector. And while I was in grad school, there was a free lunch offered by McKinsey,
Nice.
where they basically held information sessions for folks like myself who had at the time absolutely zero business training, and helped explain, how essentially a background that was deeply scientific could actually be useful and applied in a McKinsey context.
Hmm.
So I was invited to apply again, free pizza goes a long way, I guess. And I actually spent about three-ish years out of grad school at McKinsey, learning the basics, understanding essentially how the industry actually works. In grad school, and this is perhaps just my naivete, but really didn't understand how companies actually work, how all of the infrastructure and all of the regulatory systems. I mean, at a very, very high level, I knew that was a thing. But really, McKinsey taught me the basics around how does the industry work? What does it mean to actually take a drug from a concept all the way to commercial? What are the commercial considerations? What are the manufacturing considerations? These were all brand new to me and very interesting and exciting. Um, but I'll admit that I did feel like I had lost a little bit of my own purpose and mission that
Hmm.
I was originally so passionate about. And decided to zag and really kind of leapt into the unknown a bit. Moved to Kenya and really got deeply excited about the idea of contributing with my own two hands and eyes out in the field. So I joined the Clinton Health Access Initiative, which is an NGO that's focused on essentially providing support to the ministries of health. So a lot of the McKinsey skill set I had developed could be applied in that context. And uh, it goes by CHAI for short. CHAI actually is essentially composed of two major types of teams:
Hmm.
local country teams that are composed of locals that really sit side by side with folks within the ministries of health and help them think about what the programs need, the strategy, the logistics, and really supplement those teams, as well as global teams that focus on a specific domain like HIV or vaccines. And those teams cut across multiple country teams to support them from either a tool or a macro insight or strategy perspective, and then dive in to support the individual country teams that might need an extra boost,
Hmm.
or even just to share learnings across countries. So I was part of that global vaccines team. And we covered a portfolio of about 14, 15 countries. I also led our expansion from sub-Saharan Africa into Southeast Asia. And, it was an incredibly humbling and formative time. So I spent a lot of my time working with the ministries of health at the capital, but then also going out, truly out there in the field, hundreds of miles away from the nearest city, and observing what was truly happening in vaccine campaigns in rural areas, both from what healthcare providers were doing and what education they were providing to the patients, but then also logistical things like what does the cold chain actually look like? are the vaccines actually still viable when they're being administered? What is how are we thinking about the needle-based waste, et cetera, et cetera. And this was a super interesting time in that across the sub-Saharan African countries that are supported by Gavi, which is a major NGO focused on, it's the essentially also part of the UNICEF and WHO community. We were in the process of switching from the oral polio vaccine to IPV, the kind that we actually use today, and is actually going from an oral drop to an injection. We were in the process of rolling out rotavirus vaccine and also the HPV vaccine, which was particularly interesting because we were actually looking to vaccinate girls that were roughly the age of 8 to 10, as opposed to babies. So it had a completely different logistical need and approach. So it was fascinating. It was incredibly humbling. It really helped me cut my teeth in operations and understanding what it takes to truly deliver vaccines to millions of people. But it also opened my eyes quite a bit to think about what my personal contribution could look like. And to maybe bring it to life and tell you a little story, I was part of a field team out in India in Madhya Pradesh. And, we talk in vaccines, we often talk about the missing fifth child because we have pretty good vaccination rates for about 80% of children usually. But it's that fifth child, that 20%, that somehow always falls through the cracks. And so we had been out in the field. A woman had just given birth, and we were checking in to see whether the baby had been vaccinated with Hep B and a few other things, asking her about her plans. But she was actually surrounded by a number of her older children. And as she was leaving the clinic and walking away, we started asking through a translator, how many of these other children were vaccinated, because they were certainly of age where they should have actually been fully vaccinated. And it turned out that, two of her older children had not been vaccinated at all, and they just kept walking out the door. And my heart sank, and I thought about, wow, here they are. These are the missing kids. They're walking away. And it still felt to me that while the fieldwork was very illuminating, that perhaps, in fact, the biggest way to actually impact catching those children was actually going to be at a systems design level, and perhaps even more at a funding level. And that drew me back to thinking about, okay, where is the money? Who is making the strategy? How do I actually make sure that I'm high enough in the funnel of where I can have an impact? And it brought me back to the US and McKinsey, and eventually to other zigzags. But it really did help me get grounded in, a lot of humility, a lot of deep respect for the folks who do that field work, but also thinking about where I could personally have the best contribution.
Mm-hmm. Absolutely. Absolutely. And I mean, and again, we'll get to Lilly in a moment, but one more zigzag that I really want to touch on, and because I think it's important, is that you next, following McKinsey now at this point, you become an entrepreneur and a found Santa Bio, and this is, it's sort of described precision immunology, but this is like immunology 3.0 or 4.0, whatever the dot is. I mean, we're talking antibody drug conjugates, bispecifics, selective protein degraders, really advanced stuff, advanced payloads. And, I'd love to just, if you could share your entrepreneurial experience again in setting something like this up. But at the same time, when we're talking about some of these themes, can you say a few things about, when you're on sort of the bleeding edge, as I say, with, in this case, the next generation of antibody development, is there's not a specialist for everything, I guess, that knows this stuff. Clearly, there's resources in Lilly, we'll be getting there. Um, what was involved, I guess, in putting together, the team that, could work on the bleeding edge of something like this? And then did AI and ML, which we'll also be talking about when it comes to, again, designing these multi-omics targeted antibodies, play a role at this point in the career, or not yet?
Yeah. I guess the short answer is yes. So kind of fast forwarding into Santa Ana, I had worked with Peter Emtage, who's the CEO of Santa Ana Bio, in my McKinsey life. And we got along super well, and we had done a number of different strategy projects together. And it felt like we were really on the same page in terms of where we wanted to start evolving, um, either the mix of cell therapy or precision biologics, and really excited about what we could do together. So as I was winding down at McKinsey and talking to him a little bit more about, what he was actually already an EIR with Versant Ventures. We started, continuing to play with ideas, and got excited to kind of jump in and go for it. So yeah, the experience of building a company from absolute scratch was incredible. It was literally everything from back office, like we had nothing, right? So, what do we need to set up in order to hire people? What do we need to set up in order to operationalize a lab? But then obviously most interesting and exciting was, how are we going to have a truly differentiated program? And this is also part of where we met our other co-founder, Danny Wells, um, who essentially brought a lot of exciting bioinformatics background to the table. And we started playing and pushing further on what our program concepts would be in terms of where we wanted to focus essentially our lead program, and leveraging a lot of what Danny had developed on the platform side and the bioinformatics side, Peter's deep knowledge of immunology, and really marrying those into also pushing my perspective on what differentiation and strategy would look like for a biotech to be successful. So it was a really great, three minds kind of coming together with different experiences and different perspectives. But I think we got to a place where we were really excited about program number one, program number two, program number three, and it became a really we were very fortunate to get quite a bit of funding to be able to start making those real.
Absolutely. It was a very impressive raise at the same time. So hat off to you on that one. Okay, so yeah, I mean, let's work now towards Lilly, and as you probably know, I've had the honor of profiling several of your crew over there over the last couple of years that have each talked about artificial intelligence and machine learning from sort of different angles. So Andrew Adams from sort of the Genetic Institute that you have there, Diego Rao sort of more broadly from sort of the operational nature of the company. Reintroduce, if you would, sort of where Eli Lilly currently sits in terms of sort of the AI machine learning spectrum across the organization, because obviously we all want to be at some point, it's where I push a button here and the perfect pancreatic cancer drug pops out there, but we're not there yet. But how do you view the AI and ML in 2025 across the Lilly organization?
Yeah, I think so. I would say with confidence that AIML touches 100% of our programs.
Okay.
But then I guess the real question starts to be to what extent, and how influential is it over perhaps wet lab data, for example, in terms of decision making. And I think that's where we have, still, and this is just an industry wide, it's not really Lilly specific. But this is where I think we have the most opportunity to continue growing and gaining. So, if you think about our vast history, particularly on the small molecule side, for example, we have already been using property predictive models for at least 15 years. In every program, as just a matter of this is the workflow, right? And a big part of it is actually running it through property predictive models. As we venture into our antibody programs, we certainly do that now also as a matter of course, but obviously that's newer. And I think just generally speaking, probably if you think about it, small molecule, we have decades worth of data. Antibody, we have years of data, and then if you think about genetic medicines, that's where we're still sort of building the as Andrew would have described, we're still sort of building the infrastructure, and very actively collecting data, but certainly where we have the least today. And so really, I think, our ability to leverage AI is probably commensurate with those aspects of how much training data do we have, therefore, how much are we able to do in silico in any given modality, and how much can we rely on it for decision making. So these are all interrelated, and happy to extrapolate on this, but a big part of what we're excited about in launching Lilly TuneLab is essentially really being able to both bring to bear what Lilly already has and does, but also essentially continue to expand it and push the boundaries together with the ecosystem.
Mm-hmm. Yeah, before we touch on the TuneLab, let's talk about the broader ecosystem here that you're developing per Lilly Catalyze, and again, it's been very interesting. I've hung out in this industry for the past couple decades, and I remember the days where it was just internal research, and then it was internal research plus business development, then it was the internal venture fund, then it was the venture philanthropy,
Yeah.
and they each played a specific role in sort of how early on we'd like to invest and we think about these different technologies. It seems with Lilly Catalyze, you're sort of bringing together the best of all worlds in the sense of combining the ability of a pharmaceutical company, venture funding, sort of CRO thinking, and then now with the AI, the sort of tech bio brilliance, all into sort of one package that you're offering to the industry. Why did you, just in general, introduce Catalyze360 and why Lilly decided, okay, this is a new approach that we are going to go after? It's a really great approach, but just talk about a little bit the history.
Yeah. No, happy to. So Catalyze360 is essentially our external innovation team, and we certainly probably have a broader offering than some of our peers, but it is essentially our equivalent focused on interacting with the biotech ecosystem. So we have 4 pillars. We have our Lilly Venture team that actually focuses on making early stage investments, and this is really all about essentially finding the best science wherever it lives, and supporting it in this instance through funding. Lilly Gateway Labs, which is our innovation hubs, where we both provide state-of-the-art space in a modular way so companies can actually grow with it and not have all of that capital expenditure at the outset. But also creating community, creating access to expertise within Lilly. So these are our friends and family companies, we like to think of it.
Sure.
And Lilly Explore R&D, which is relatively new, a couple years old, but actually grew out of an offering that we've had for many decades called Chorus. Essentially, Explore R&D provides R&D services to our biotech partners, everything actually from preclinical all the way to actually supporting clinical trial work. And so this is where Lilly expertise can be brought to bear to essentially continue the development of a program that a biotech owns. And then our latest Lilly TuneLab, which we just launched a month ago, which is in my mind it was clearly the missing piece. So we had everything from capital to innovation hubs to services. We weren't yet really offering anything that was in silico focused. And we had been asking ourselves, in the new era of AI, which I guess is still a relatively new phenomenon, how could we think about both supporting but also collaborating and partnering with companies in a completely different way? And so that is where Lilly TuneLab came from.
Mm-hmm. Yeah, and let's drill down a little bit more on TuneLab because you talk about it in the public material. You've to date put somewhere around $1 billion alone into all things AI and the discovery process, and clearly, a company like yours, which is whatever 150 years old now, you clearly, whether it's toxicology data or ADME or pharmacokinetics or all the other stuff data-wise that you have in-house,
Yeah.
You've no doubt trained those AI models on a lot of great data that not a lot of other people have.
Right.
Drill down a little bit on that, of what it means to have spent $1 billion on AI research to date, and a little bit specifically of what TuneLab offers. If hey, I come along and I want you to whatever run a model on such
Yeah.
and such data for me, and how that all works.
Yeah. So it won't be a surprise to anyone that we have, again, decades worth of data. So that's where the billion figure comes from. Essentially, it is truly all of the proprietary program related data generation that we've been doing to advance our portfolio. So this is sort of the base training data that we're leveraging in TuneLab. Actually, to be clear, to date, we actually don't even use the public data sources. So this is truly all proprietary data. I think one thing that's particularly exciting about our training data is Lilly actually, from the start, had the pretty incredible foresight to think very carefully about protocol drift and data harmonization. And so there's actually very clean data here, both in terms of exactly how it was collected, but then as protocols shifted, really thinking about essentially what controls we could use to harmonize across those data sets, and clearly demarcating if there was a big departure. For example, a new instrument became available, that we actually treat those as completely different data sets. So we have a lot of just really clean data to use from a training perspective, which is unique. And a big part of what we wanted to think about, and I lean on my experience from Santa Ana and other kind of scrappy biotechs that I used to work with all the time, is really thinking about essentially how could we really better accelerate the ecosystem, because there is so much that lives in our walls that, again, we do as just part of our routine for any program, but that these companies essentially don't have the data sets to create their own equivalent models. Mm-hmm. And frankly, it would almost be a waste for them to even focus on that. I would think about, and again, I'm leaning on my Santa Ana experience, but if you think about differentiation when you're a biotech, it's probably going to be primarily on a novel insight that you have on the program's design, whether it's novel biology, you're thinking about an exciting new target, or you're thinking about an interesting way to modify the format of an antibody, for example. But it's probably not going to be in, okay, how am I going to run my ADME assay? Or how am I going to think about measuring viscosity of this antibody? And so what we really wanted to do was think about, okay, everything that's sort of downstream of, you've made a decision that you want to create a program here and you have a hit, but now you have to go through the steps of essentially getting all the way from there to a development candidate. How could we really lean into that and essentially provide the efficiency that Lilly has created internally and make that available to our biotech partners?
Mm-hmm. Awesome. And in terms of those biotech partners, I mean, you have dozens of companies that have joined in the system, and it's something like 50 plus different therapeutic platforms being developed currently. Your companies in the system have raised upwards of $2 billion. Again, I mean, I know there's a lot of public material out there. Obviously, a lot of it's private, but whatever you could talk about in terms of some examples of sort of the type of partnerships that you've facilitated, the types of companies you're working with? And again, don't go into anything that I have to edit afterwards, but take us down that path on some examples that you're allowed to talk about.
Yeah, I mean, what I can definitely share publicly is so at the moment we essentially have two archetypes of collaboration within the TuneLab realm of the world, right? We have companies that are participating in the platform. So it's designed to be a collaborative platform. So we give companies access to our models, and in exchange for that access, we get a training data contribution. So this is a completely zero cost partnership value proposition to our biotech partners. When they join us, they do commit to making a training data contribution. And so in that realm of the world, that's archetype number one. This is where, you know, we currently have over a dozen partners. As we've launched, we're in the process of onboarding quite a large wave of new participants. We certainly aim to get to, you know, triple digit number of partners that look like that. So that's one major archetype. I would say the second major archetype is looks more like our partnership that we've announced publicly with In-Sitro, where we're actually collaborating with them to build and then eventually make available on the platform new models. So the ones that are available today are the same exact models that we already use within Lilly. But we want to continue pushing the envelope on what's possible. And in this instance, working with In-Sitro to actually develop in vivo property predictive models of ADME and PK.
Got it. Along those lines, now, if again, if I was Dr. Aliza Apple, I'm come along, hey, I got a really great idea for a minimally invasive intervertebral disc therapy that I want to develop. I have some initial data. I mean, what's the how do you start the process off? Do I have to show up with like my Series A round already in place, and some animal data? Like, where do you sort of what's sort of the portfolio preferences if this was, I'm looking at this like sort of venture capital firm, like, nah, we'll see you in a couple of years, come back, versus, you know, who you allow in, sort of what's the early stage profile of someone that's allowed to at least come have a meeting?
Yeah. So actually, we're being very purposeful that we are open to working with companies of all stages, whether they're pre-seed or we've even, been talking to some mid-sized pharma companies that are excited to join us. So truly full gamut of pre-seed and very preclinical to actually already commercial stage. The most important criteria from a practical perspective is that they essentially have active preclinical programs that relate to the two use cases that we already have live on the platform, which are essentially whether they have a small molecule program or they have an antibody program. We do aim to expand the number of modalities that we have use cases for over the next year. But with that in mind, it's really more about two things. One, ensuring they're philosophically aligned with the give-get model, and actually, we do put agreements in place to govern the training data contribution that they would make. And then the other is we do, of course, some, the equivalent of like a background check, if you will, to ensure that it is a legitimate company that has funding that's actually likely to actually be able to generate the data. Because we actually do not make it a prerequisite for them to have training data to contribute out of the gate. Okay. So again, kind of leaning on my own personal biotech experience, but also we've done quite a bit of, a listening tour, if you will, across our portfolio companies to better understand what would be useful. There are plenty of companies that would be excited to use TuneLab, use our models, but that probably will be generating a lot of data downstream from the use of the models. And so our data contribution is actually essentially designed so that companies can, as soon as they execute the agreement with us and get started, they can actually start using the models right away, we ask them to make the training data contribution over the course of their first year with us. So a lot of companies, if you think about the workflow, for example, will actually probably use the models upstream, actually use them to decide which molecules to even synthesize, and then synthesize the molecules, then do the wet lab experiments. And that's the point at which they'd be able to make a training data contribution. So we didn't want to put the cart before the horse and say, in order to join us, you have to have training data in hand. We actually wanted to make it broader and more approachable and easier for early stage companies to participate, because that's a big part of, frankly, who we've designed for here.
Sure.
And make it possible for them to participate, actually get the value on day one and start using the models right away, and make their training contribution later when they would naturally have generated the data.
Got it. No, it's a really exciting model. And again, you're bringing together now with the AR part of it, the complete package for for a lot of early stage folks. So it's exciting. What at least what else is coming up that we should know about? I mean, I know I see that you get out on the conference circuit quite a bit, and talking about the 360 model, and now of course TuneLabs, but conferences, talks, other public-facing initiatives per your organization as we head into the end of 2025, the early part of 2026 that we should know about, or anything I missed while we have you today, please.
Yeah, I mean, yes, I'll be on a panel at Health. We always do a Lilly, event at JP Morgan. We'll be participating in Bio. So yes, for better or worse, we do definitely do the conference circuit. I think in terms of what you might expect from us, announcement wise, we are continuing. So our collaboration with In Citro was the first of hopefully many that will look like that. So we're actively shaping a number of partnerships to build new models across different modalities with new partners that I'm hopeful that we will be able to announce at the various upcoming conferences. So that's all coming together. And, I think a big part of the value that TuneLab brings back to Lilly, of course, is improvements around the models themselves with a lot of exciting new training data. But also, this is a new way to think about partnerships. It's a new way to continue to push what models can do. And so a lot of, I think, what we're also able to do at the tip of the spear for the broader Lilly organization is think about who should we collaborate with, and what can we learn from those much more technology focused collaborations, much rather than, a lot of the existing collaborations that we tend to do are more asset development oriented.
Sure.
And the ones that my team is leading are much more focused on, essentially developing new technology. So that's a relatively new and certainly very interesting and exciting development for Lilly.
Mm-hmm. Yeah, it's quite an exciting set of initiatives. And again, I'm sitting here, thinking you took us from regenerative medicine to global public health to antibody engineering, and now into this really awesome, novel ecosystem, and that's just not existed. So I'm excited to see how this all continues to evolve, and you move all these types of programs forward. But it's very exciting. Again, for everybody out there that's going to be listening to this episode across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Aliza Apple, Vice President, Catalyze360 AI, Global Head, Lilly TuneLabs at Eli Lilly, doing really amazing work to develop these novel innovation ecosystems to, as they say, accelerate both emerging and promising science. Aliza, I want to thank you for taking the time to sort of journey down memory lane on where you've been, where you've headed, and where you're going with this. It's really impressive. Obviously, thank you for everything you do to gestate and develop these biotech companies, as we like to say on our show. Thanks for helping to create a better tomorrow through what you do. Really an awesome set of stories. I really enjoyed having you.
Thank you. Thanks so much. It was a pleasure. Appreciate it.
Great having you.